Replication of DNA is when you copy that DNA. It's a healthy, normal process to make a whole new copy of that DNA. We're going to look next at, instead of replicating DNA, how to take DNA and turn it into a protein. And there's two steps in that process. So if I have DNA, first I'm going to turn that DNA into mRNA, and that is transcription. And then the mRNA, with some help from tRNA and rRNA, can be translated, translation, to turn it into a protein. So we're gonna look at transcription over here from DNA to mRNA. We need that because this is all gonna happen in the nucleus. We don't want our DNA leaving the nucleus. So this will happen in the nucleus. And then our translation, turning it into a protein, that's going to happen in the cytoplasm. And you've got some videos to watch that bring this much more to life than I can on a page here. So I'm just going to give you a few terms and examples here, and then make sure you watch the videos of transcription and translation that I provided for you. So taking a look at transcription here, if I start with a DNA strand with these letters in it, transcription is going to be where a small segment of your DNA, we don't want all of it, we just want one gene. So we're going to take one gene and we're going to take that DNA and turn it into mRNA. Notice here the DNA is double-stranded. mRNA is just going to be one strand. So we want to make this tiny little strand that can leave the nucleus. mRNA is going to have opposite directions, so 5' to 3'. And I want to transcribe it with RNA. Our bases are going to be C and G, which will pair up, and then A and U. So the T gets replaced by uracil, the thymine gets replaced by uracil, and so the A's will pair with U's and the C's will pair with G's. So as that my mRNA comes in, and this would be my mRNA strand right here, my T will pair with an A, C with G. A now will pair with a U because it's RNA. C, G, U, A, C, G. So this would be transcription on a tiny scale. It would be much longer than that, but a tiny scale. I just transcribe those complementary bases, and don't forget to use your U's there. So transcription is going to be that synthesis process, and now there will be some editing done of this mRNA before we go to express it as a protein. So I'm going to take this and I'm going to definitely do some editing that is way beyond the scope of probably all of us for all of our lives, but a lot of scientists are working on that. And then we're going to send that mRNA strand over to the cytoplasm where we can do translation. So just to kind of see what's happening with the bases here, you don't have to think about all of this every time, but it's good to see kind of the progression. So DNA, again, this would be in the nucleus, and we're gonna just grab the DNA from one gene. It gets transcribed over here into mRNA. So we already did that, AGU, CGU, ACG. So this was transcription. All right, now the mRNA here's my mRNA, comes to the ribosome, and as it feeds through that ribosome, there's 3 spots in the ribosome where these 3-letter codons are read. And so mRNA, you can see we kind of break it down into these groups of 3. It's kind of how it's written because that's how our ribosomes read our DNA. Or our RNA. So you'll notice here as the mRNA comes in, it gets some help from what's called tRNA, and the tRNA molecules are kind of in the shape of T's here, all right? Each tRNA is going to complement what that mRNA has. So if mRNA is AGU, the tRNA is going to match up with a UCA. Notice that here. So here is the AGU, and the tRNA has an UCA. Now, what's really exciting about that is this tRNA would have had an amino acid connected to it, and I have lost track of what that is, so I have to come down here and actually read with this codon table. All right, so this codon table is always going to be read from 5' to 3 prime, so make sure I'm reading 5 prime to 3 prime. And I want to look at the mRNA, AGU, find it down here. So here's A, second letter is G, so here's all my AGs, and here's my AGU. So this tRNA would have initially had a serine on it, and this amino acid then right here is serine because that was just added. So my protein would have a serine in it. CGU, here's C and G and U, that's arginine. So this amino acid here is arginine. So what tRNA does, they're called transfer RNA. They're in the shape of a T. They have this anticodon here, and then they also bring an amino acid to the ribosome, and those amino acids get linked together until I have a polypeptide or a protein. And again, the videos will bring this to life for you, but you'll see the little tRNAs keep showing up, matching up to the mRNA and translating them into proteins. So we translate genetic code into protein code is essentially what's happening here. So CGU was arginine, and that was this GCA. These are the anticodons. And then finally here, the one that's about to be added on is ACG. So ACG is threonine. And that anticodon is UGC. So here's my protein, and then my protein will go and fold up into, we're already building the primary structure. So this is my primary structure, but it'll go fold up into secondary, tertiary, quaternary structure, and then go off to whatever function it's going to have. And that is the goal of translation, is to generate that protein. So transcription happens in the nucleus, and we get a little strand of mRNA. mRNA comes out of the nucleus to the cytoplasm and feeds through the ribosome, which is made up of rRNA. So I have this teamwork between mRNA, rRNA, and some proteins, and then here I have my tRNA. So all three are working together to synthesize this protein. Notice that I drew bonds here because those amino acids are actually literally bonded together, right? So here's my threonine, arginine, serine clicked together. And this will be roughly on average 400 amino acids long, and it'll start to fold up. So just a couple more terms as we finish up the translation here. Initiation is how you're going to start your protein synthesis. And that is always going to be with a codon AUG. So every protein starts with a methionine. These are those same amino acids we learned about in chapter 16. So we'll have that, and it often gets edited off at the end of the protein synthesis. Elongation is when you just build all those amino acids in the middle. So you keep adding amino acids to build that protein. And then termination is when we say, okay, that's it, that's the end of the protein. And so to terminate, you stop, and you can see that there's some stop codons here. So we want to use one of those stop codons. So when a mRNA shows up that has one of these stop codons in it, maybe say UAA, it would not bring an amino acid, and so that would effectively stop your protein production. So that's transcription and translation, and be able to also compare those to replication.